Hi everyone, welcome to the latest episode of RNA sequencing for beginners. Today we're going to be talking about aligning and quantifying raw RNA sequencing data using the platform called Illumina DRAGEN. There are a couple different types of tools you can use to quantify RNA sequencing data, but at least within our lab, we use Illumina DRAGEN because it's fast and ultimately leads to quicker biological insights, and ultimately we think it's worth it, even though it costs a little bit. So with that, today we'll be going through the steps of RNA-Seq sample preparation, raw data output, what actually is, what it means, and how you will perform the quantification and what quantification actually is. So let's start with how RNA sequencing samples are prepared. So looking at the typical preparation of RNA-Seq samples, you all are probably all familiar with RNA extraction. So pick your favorite kit, and RNA is extracted from your sample of interest. But the thing is, you have a lot of different types of RNAs in the actual RNA extracted material. So first we need to perform a poly-A enrichment. And the way we do that is using these beads that have these oligo DT sequences on them. And this will essentially pull out all of the RNAs that have a poly-A tail on them. And interestingly, actually the majority of RNA in a cell is ribosomal RNA. So this is getting rid of all of that, as well as a lot of other different types of RNA, such as tRNA, SNO-RNA, small nuclear microRNA, and long non-coding RNA. So we really just want to focus on poly-A coding RNA. And so next, once we've enriched for these poly-A RNA molecules, we then want to convert these single-stranded RNA molecules to cDNA using reverse transcription. And the reason why we're doing this is that sequencers are built to use AGCT, and RNA has uracil in it, so that just makes it difficult to work with the technology. So we have to convert cDNA for that. Also, RNA is very unstable, and so through this process, we actually remove the hydroxyl group from the 2' carbon of the sugar backbone, and this makes it more stable. Next, once we've done that, we take these single-stranded cDNA molecules and we prepare libraries by adding adapters and indices, and then we amplify these fragments up. I'll probably make another video on library preparation in general, but I just wanted to cover at a super high level what's actually happening with your RNA samples. I will also note that a lot of sequencing cores now offer this whole process in-house. So really, for the most part, you shouldn't really have to do this yourself, but it's good to understand what's actually happening so you can interpret the data better. And so then next, in this imaginary sequencing run, you send your samples to a sequencing core, and they will return to you some sort of raw output. So just to give you an idea of how we typically interact with these sequencing cores, we will request to have our samples sequenced paired end, meaning it's sequenced from both ends of the DNA fragment, and we want 20 million reads per sample. So here on the right here in this image, I've kind of shown a crude illustration of what your RNA sequencing libraries will look like. So you have your DNA insert, which is the regions you're actually interested in, but flanking the DNA insert, you have these adapter and indices on either end of this. And so when we say paired end, we mean that the sequencer is going to be reading this DNA insert from both sides. And so that's the paired end read part of it. And ultimately, because it's sequencing the DNA inserts from both sides, you're going to get 2 data files for each sample. Here on the bottom, I'm showing you what the files typically look like. They will either have the suffix FASTQ or .fq. And so those are kind of the traditional raw read files. I guess if you want to take one step back, you can take BCL2 files, but typically the sequencing cores will give you FASTQ files. And just to kind of conceptually understand what is in each of these files, all of the reads in the R1 FASTQ file are all coming from this one particular end of your library. And then in your R2 FASTQ file, all those reads are coming from this end of your particular sequencing library. And each of these files has 20 million reads respectively in it. So just trying to kind of cover what this terminology means in terms of paired end and how many reads you have per sample. But ultimately, we're going to use these two FASTQ files for each sample to perform aligning and quantification. And then next, once we've obtained the raw data, we move on to the quantification step. And in this case, we are going to be aligning these raw reads to a reference genome. In our lab, we tend to focus on GRCH38 P13. The reason for that is this is the most commonly used reference genome, and we kind of picked one and just stuck with it because all the downstream analyses, it's really important that you use the same reference genome. If you start trying to use different reference genomes down the line, things can get pretty wonky in your analyses. And then next, after we align it, this alignment process helps us figure out which part of the genome each read corresponds to. And then we count the number of reads that map to each gene or transcript. This count gives us an estimate of gene expressions. So basically, from a high level, the more reads that are aligning to a particular gene, that indicates that there's higher expression of that particular gene. And on the right here, I'm showing you what a example output from DRAGEN will look like. And so you have these 5 columns. In the first column, you have the name. And so in the case of DRAGEN, it's actually quantifying the number of reads that align to each particular transcript. And like I've said in other videos, there are multiple transcripts that correspond to one gene. And so later down in the analysis, and if you want to go look at the DE-Seq2 step-by-step, that'll show you how we're actually aggregating these transcripts for the gene level. But ultimately, I just wanted to show you an example of what these quantification files actually look like. So now that we've covered the basics of RNA-Seq sample prep, raw output, and quantification, next I wanted to show you how we're going to obtain this reference genome, and then we'll jump into the actual steps that are required to perform quantification using Illumina DRAGEN. I just wanted to do a quick disclaimer of the requirements needed to do the analysis today. Instead of earlier RNA-Seq tutorials, we will not be using R today. The only language we will be using is Unix. The only things you'll need to do this analysis is an AWS account, and you will need access to Illumina DRAGEN. This pre-made Linux instance costs about $20 an hour, so be prepared for those costs. But once the analysis is done, we'll move the data from this instance onto the S3. We'll download it from the S3, move it to our studio environment, and that is where we picked up in the first video of the series. So the first thing we need to do before we begin this analysis is actually download the reference genome and annotation file for the human genome. And so where we're going to download this is the GenCode website. And so we're going to use GenCode release 36. This is the GRCH38 P13 reference genome. This is the most commonly used genome across pretty much all human research. So that's what we'll be using today, just to be consistent with what the field does. There are 2 files you'll need to download from this particular website. First, you will need to download the GTF file, which is the annotation file for the human genome. This is just telling you where all the genes are, at least with predefined regions within the genome. So we'll download that. And then the second file we need to download, if you scroll down here, is the actual FASTA file. So this is the linear string of letters that is the human genome. This corresponds to this GTF file. So this GTF file is annotating where in this string of letters the genes actually are. So we'll download that. This will take a bit to download, so I will let this finish and I will see you in a sec. So once these files have finished downloading, we need to upload them onto the AWS3 bucket, and this will allow us to move them onto our Dragon instance. So the way that we'll do that is we'll come here, log into our AWS accounts, and then we'll navigate to the S3. And I've already created this folder here called tutorial. I've already uploaded these files, but I'll show you how I did this because it will take about 20 minutes for them to actually upload. So you click upload, you hit add files, and then you will click these two. And you click upload. Since I've already done it, I'm not going to upload them right now, but you can see how this can be done. So here I will be showing you how to launch your Illumina DRAGEN instance. So you'll come to your console home after you sign in, go into the search bar, and just type in DRAGEN. Scroll to the bottom of the list to find the marketplace, and then click DRAGEN Complete Suite. View purchase options. And then click launch your software. These interfaces change periodically, so it might look a little bit different on your end, but you'll click this and you'll ultimately get to the configure this software option. Within our lab, we have found that 4.2.4 has worked, so we've stuck with this version to avoid any issues, but feel free to change versions. The only thing is that if you drop below 4.0.3, you will need to use a different hash table, and I'll talk about that a little bit more down the line. But for this analysis, we're going to be using 4.2.4. We'll keep this the same, keep this the same. We'll go to continue launch. And these are some Emory specific settings. So if you are working in another university, I would talk to your IT department to see exactly what you'll need to actually launch this instance. But in the case of ours, we have to use this particular Emory IP security group, and also we need to select a key pair. Like I said, I'll probably create another video just talking generally about AWS, but in this case, I'm assuming you have a key pair. I'm assuming you can use AWS already and launch these instances. But those are the only two that you will be changing, and I've found that the F1 4x large is plenty large and has enough compute to actually do what we need to do today. So once we're done with that, we click launch. And now the instance is starting to launch in our EC2. So you'll come back to your console home, click EC2, go to instances, and if you look at all the instances, you can see that it is pending its launch. So if we click refresh, it may take a second to actually begin. Funny story, I actually don't know if this is called Dragon or Dragon. In the lab, we've always just called it Dragon or Dragon, but apparently some people call it Dragon. So you can call it whatever you want, but for the rest of the video, I'll be calling it Dragon. So then what you'll do is then come here, click connect, and then this will bring you to this connect to instance option. I wanted to give a quick PSA to any of those who are in the Hong Lab that might be watching this, or if this is just how your AWS is set up, this is FYI to you. You will not be able to move data between your S3 and your remote computer unless you change the security option, and you have to modify the IAM role. You need to allow this EC2 to S3 access. If you do not have an IAM role, the transferring of files will not occur. So I just wanted to point that out before we get started with the rest of this video. Okay, so once you've launched your Dragon instance, you need to connect to that virtual computer with this SSH command. So this is what the suggested SSH command is. It is actually not correct. So if we copy this and then come to our local computer, this is the terminal that you can open up by just searching terminal. That's what I've opened up here. I've now navigated to the folder in which I have my PEM key that is associated with this instance, G. Cooper Hong Lab. Then I'm going to paste this SSH command, but we have to do a few modifications. At least within the Emory network, we actually have to change the IP address to this private IP address as opposed to the public. I don't exactly know the details, but that is how we have to do that to get into the instance. It may be different at your particular institution. The other thing we have to change is we have to change root to CentOS. And this is actually a very common error when you try to connect to this instance. So it's going to give this remote host identification has changed. And so all you'll need to do is copy this IP address right here, SSH keygen R, and then type that in. To be frank, I don't exactly understand why this happens every time, but it does. So I have to do this almost every time I launch this instance. And now when we run that SSH command, we are now into the instance and we say yes. All right, now you can see this CentOS kind of header has changed. So we're now accessing this virtual machine that is pre-installed with Dragon. You can see that there's nothing in this anymore. I'm no longer on my local computer. And so now what we need to do is navigate to this directory called ephemeral. This is a little strange in that the actual compute power and storage is only in this ephemeral directory. So I'm navigating up 2 directories and down into the ephemeral. And there's nothing in this one, but you have to navigate to this directory for this analysis to work. So now that we've navigated to our ephemeral directory within the remote Dragon instance, we need to move our reference genome and annotation file onto the ephemeral directory. And so the way we're going to do that is navigate to this folder that we uploaded our files to the S3 in, and then you have to copy the S3 URI, and then in the ephemeral directory we will type in AWS3 sync, and we're going to be syncing the tutorial files with the current directory. And this should be relatively quick. And so we have our files here. And now we need to unzip these files so they actually are readable to the Genome hashref software. And then GR. This one might take a little bit more time. Yeah, so right now what we're going to do is we're going to take these reference files and create a hash reference. And what this hash reference is a special indexing file for the reference genome that Illumina uses to create very fast alignment. Typically, alignment can take quite a long time using other tools, but DRAGEN's really fast. It only takes a couple of minutes actually to align the genome. And so this hashref is really what allows DRAGEN to do this. And so now that we have uploaded our reference files and unzipped them, we need to make 2 directories. And then one called hashref and the other called ref. And then we're going to move our reference files into this ref directory. Nope. And we'll move our FASTA file. Just make sure they're in there. Yes, they are in there. And so now we're going to run a command that actually creates this hash reference. Okay, so now we're going to run that command like I just said. That's going to make the hash reference. So I'll navigate to this R markdown file that will be shared on GitHub, and you can copy this command yourself. So we'll copy that command here. This is really, like I said, just building a hash table and pointing it to the reference genome and the output directory. One thing I will say is this says enable CNV true. This will come in handy down the line when we do structural variant calling, and this reference can be used for lots of other analyses as well. Right now we're going to be using it just for RNA-Seq quantification, but you can use the same reference genome to perform structural variant calling, variant analysis, attack-seq alignment. Basically, this reference genome is good for most genomic analyses, so I just wanted to include that so we can use it in the future. But yeah, this part will take around an hour to 90 minutes to actually finish. So I'm going to let it do its thing, and I will see you when it finishes. Okay, so it looks like our reference hash indexing has completed. This particular runtime actually took an hour and 47 minutes. So when you run this, just take into account that it's going to take a little bit of time. So now we can look at what the hash index actually looks like. And so here, if you go on the hashref, you can just see what our input files were. And then you can go to the ref folder, and you can see that this is the hash index ref here that we'll be using to perform alignment and quantification. So I always like to just have these files in the hashref or in the ref directory, just so I can move everything onto the S3 so I can use it in the future. You also will need the GTF file in the ref directory to perform the actual quantification. So I'm just going to move these to the ref file, or ref folder. And now you can see that we have our GTF file and our FASTA file in this ref hash index folder. And then in the future, if you want to use this hash index again to perform alignment, I just move it onto my S3, and then you can basically keep it there as a saved index, and then you can always just move that directly onto a new DRAGEN instance, and you won't have to be doing this hash indexing every single time. So you will not need to wait an hour and 47 minutes every time you try to align and quantify a genome. You just move this onto the DRAGEN instance and proceed with what I'm about to do. So to show you how I'll move this onto AWS S3, if I come over here, here's my tutorial folder. I will make a new folder called grch38p13 version 9. I'll navigate to there, copy the path, and then I will sync all of the files in this particular hashref index and move it there. And so this is quite a large file actually, so I'm going to let this finish syncing, and we'll see you when it finishes. Okay, so it looks like it finished transferring these files in our Href index onto the S3 bucket. And just to show you what that looks like, all these files now are in our S3 bucket, ready to move back to a new Dragon instance if you want to do this again. And just to show you how you would move it back on, I'm not going to do it now, but if I copy this path here, come back to our instance, and say you wanted to move it onto the new instance, I'm not doing it now, but just to show you, you would say sync everything on there and then put it on there. So there's directionality into how these sync commands work. But that would just move everything back onto this instance, and you could proceed with what I'm about to do now. So I just wanted to show you that really quickly. And so now we need to format our instance to have the necessary output folders, input folders, and let's write the code chunk to actually perform this quantification and alignment. So I'm just going to remove the Href as we don't need it anymore. And I'm going to change the name of the ref to what we have here in this code. This is what I call the reference. This is just how I always align my samples. So now you can see this is our reference folder. And this is the code chunk that's actually going to be run, that's going to perform the aligning and the quantification. And so as you can see, there's a couple different folders and input files we need to pull onto the instance for this to actually work. First, we need our input folder called RNA, and that's where we're going to put our raw FASTQ files that we got from the sequencing core. And we also have to create our output file, and I just name it RNA out. It doesn't have to be named RNA or RNA out, that's just what I call it. And so we'll come back here, and we'll make the directory RNA, and make the directory RNA out. All right, so those are there. And then I'm going to navigate to the RNA directory and pull in the raw data. So while that Href indexing was cooking, I moved on one sample from the preprint that just came out in my lab that I just aligned not too long ago. I'm just going to move that into the RNA folder. And I just use the copy command. I use the copy command when I'm moving just one particular file, and I use the sync command when I'm trying to move multiple different files into a particular folder. So that's just kind of the nuance of how AWS CLI works. And similarly, I'll move this file as well. S3 copy. And just to kind of highlight more the directionality of this command, like I was saying, the path that comes second is always where the files are moving towards. So we're taking the files that are in the S3 and moving them towards our current directory. And if we flip the direction or the order of these two different paths, the direction the files are moving will change. So just kind of wanted to walk through that a little bit. All right, and so then now we need to create our code text file. And I typically use Nano when I'm in Unix like this, but it doesn't come pre-installed on the remote instance we're using. So we actually have to use Vi, which is kind of annoying. I like Nano more, but that's what we gotta work with. So I'm just naming it code sh. You can literally name this whatever you want, but I'm just gonna name it code sh. And so then I'm gonna come over here and copy these commands that I have pre-made to align the two. I guess I only have, I'm just gonna do this one because we only brought in these two files. Another thing I've noticed when copying this that often catches beginners off, for some reason the first three letters don't copy when you use Vi, so that's just something, make sure to type in DRA there. If you wanna align multiple files, just make sure to put this semicolon after the end of the first one, and you would just copy this whole thing. And so you can expand this to as many files as you want. In this case, I'm only going to do one just for time's sake. But if you wanted to do two, you would just put the semicolon, copy the same code chunk, and just change the names of the input as well as the kind of output file prefix stuff here. And so just kind of walking through what this command actually is doing. Here we're just really calling up the DRAGEN software, and here we're providing the reference genome. These are just the input files. Here I'm just giving it the annotation GTF file, and we're just telling it we want it to map and align, we want it to enable this as RNA analysis, and we also want to enable that we want to quantify how many reads are aligning to each gene. Here I'm just saying I want the BAM outputs. This doesn't really need to be there, but I'll just keep it in case. We really only want the quant files. Here I'm just showing where we want to put the data, and these 3 lines are just saying here's the prefix I want for all the output files. And frankly, I don't know why you need the 01 and 01S1 here, but you do. It won't run correctly if you don't have it. So that's what the code is. And so now that we've input it, we need to save this vi text file. And so it's a little weird, but the way you save this is you first hit escape, then you hit colon, and then you hit X. You can see the colon and the X appear in this bottom corner. That's how you know it's working. And then you hit enter. And so now you can see that this code sh text file has been created. And so we can just look in there and make sure it's right. Yep, that's everything we put in there. And now we just need to change the permissions of this particular code file. And so the way that you change the permissions of a code file is this chmod command. This just makes a text file executable so you can run it with a particular coding language or software. And so here we're just making all levels executable. And so you can see when this happens, it'll change from a black text file to a green executable file. And before we run this, just want to make sure everything matches up. The names should be the same. P13, V9. Yeah, all right, well, let's try to run this. To run a code, you literally just do period, / whatever the code file you want to run is. All right, so now it is doing its thing. It's performing the alignment and the quantification. And I'll just let this run to let you see how this all goes. And just to talk a little bit about what the output of this is going to be. There's actually going to be a lot of output files. It's not just going to be a quant file. You're going to get BAM files, you're going to get index files, you're going to get kind of metric files of how the alignment worked. I tend to not really save these as these are very large and you don't really need them to perform basic RNA-Seq analysis. The only thing I can think of to save the BAM files for would be if you're wanting to do some sort of isoform expression analysis. However, we're only sequencing here to 20 million reads, which is not sufficient to be able to capture isoform expression. You would need to go up a little bit higher, maybe 50 to 100 million reads. I'm not entirely sure on the amount, but at least for this analysis, we don't really keep the BAM files. I'll just export the quant SF file and put that onto our AWS S3. And so you can see it's reading everything in. Very fancy. And so interestingly, when you normally align a file, it actually takes sometimes hours if you're using a traditional aligner like BWA. But as you can see, and we'll see, that this takes only maybe 1 or 2 minutes, which is so much incredibly faster and helpful when you're processing many, many samples. Within our lab, we sometimes will be aligning, I don't know, 20 to 30 samples at a time. And so if you think if that took 2 hours per sample, that can take days. And so this will take an analysis that would have taken days to do and do it in less than an hour. So I think that's really the benefit of DRAGEN alignment and quantification, as opposed to doing it more traditional routes by aligning with BWA and using Salmon itself as a quantifier here. But yeah, so this is wrapping up soon. This is the last thing I'm doing before I head out for vacation, so I'm very excited. So maybe this is taking a little bit longer than I thought. So I think I've timed this out. It's typically around 3 to 5 minutes per sample. So we might be getting there soon. If you're still here with me, and you're watching this video, maybe if you want a particular analyses type that you're unsure of how to do, leave it in the comments and I can take a look, see if I've done it before, if I can quickly look up how to do it, and I'll add it to my list of things I'm trying to post. Right now I'm focusing on RNA-Seq, whole genome-seq, cut and run, attack-seq, but I'm always open to doing other analyses, and please let me know what you want. I will try to do my best to provide help for any analysis that you all need. I guess while I'm here, I'll rant about how annoying it is that the preprint I just submitted has been under review for 50 days. So long. Can't believe it takes 50 days to get reviews back on a paper, but it does. So if you're a grad student out there watching this and you're wondering how long it takes to get reviews back on a paper, apparently it's incredibly long. So alright, cool. It looks like our alignment and quantification finished. And if it does finish well, it'll say Dragon finished normally. And now if we navigate to our RNA out, this is where all of the output is being put after the analyses. As you can see, there's lots of files. You get the spam file, all these metrics, genes SF, transcript coverage, yada, yada, yada. The only thing we really want is this. And so I'll just show you what that looks like. And this I had shown you earlier or in another video, and you can see this is the quantification for each different transcript. And so that's really all we're going to save. Everything else we're just going to toss out because we don't need it for the downstream analysis. So I'm just going to copy this file to our tutorial page. There you go. And if you come here, you can see it pops up right there. I'll then download that, and then you will move that to the directory that you want to do your DE-seq2 differential expression analysis in, and you will proceed where I began in the first video. So thank you for watching this. I know this is a little bit more advanced than the first two videos. Please leave any questions in the comments. If something's not working out for you or you're having institution-specific problems, I'll try my best to help you all. But I'm just doing what I know how to do at Emory. So that's the end of my Illumina DRAGEN video, and I'm hoping to upload another one in the future where I look at more traditional methods like BWA and salmon quantification. So with that, I'll head out, and thank you for watching.